All right, so in this video, we're going to be getting detailed about DNA replication and how it works. But before I do that, I just want you guys to make sure that you remembered what makes up a nucleotide. It's made up of 3 parts, right? So here's the phosphate, and here's the sugar, and then you have your bases over here, right? So remember that these are the 3 components that make up a nucleotide. And the one thing I want you guys to remember is how to identify your 3' and 5' ends, right? So here's your base. Like let's look down here. We got guanine over here. This is an oxygen. But when we talk about the 3' versus 5', we're talking about the carbons and the carbons that are free. So this would actually be each kink, remember, is a carbon. So there's a carbon there, carbon here, a carbon here, a carbon here, and a carbon right there. So we have 1, 2, 3, 4, 5 carbons, okay? Now, it actually goes carbon number 1, carbon 2, carbon 3, carbon 4, carbon number 5. Now, the one thing that it's a lot easier to identify with is when you look at what's on the 3' carbon and what is going to be your 5', is I like to think about it as what's available in the sense that if you look down here at the bottom, right, I have carbon 1, carbon 2. If I look at carbon 3, I have a hydroxide group or an OH that's not attached to anything. That would actually be my 3' carbon. So I would call this my 3' end, okay? And that's where it's showing you here. If you look up here at the top, let's look at thymine. I have my sugar, carbon 1, carbon 2, carbon 3. I don't have that OH. It's bonded to the phosphate. I have carbon 4. I have my 5th carbon, and I have my phosphate attached to that 5th carbon. That phosphate isn't attached to anything, so this would be referred to as my 5' end. Now, I just want to point that out because when we get into DNA replication, that definitely matters. So I'm jumping ahead, but I just want to show you on how to identify them because it's actually not too terrible when you follow that method. This is a much more simplified version, but basically there's my base. This is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Each one of these circles represents a phosphate. So you can hopefully be able to identify there's my oxygen. I have carbon 1, carbon 2, carbon 3. Notice I have it highlighted already. This is my 3' end because I have an OH group or a hydroxide that isn't attached to anything. So that's my 3' end. Now notice these two, if this is my 5' end over here, you have to kind of read it upside down. That's my phosphate. In terms of how this one would look, that would actually be my 5th carbon, my 4, my 3. There would actually be an OH here, my 2, my 1, okay? And this would be my oxygen, okay? Now, if you haven't noticed or you don't know from previous lectures, if this is your 5' end on this side, this is going to be your 3' end because DNA runs anti-parallel. So the bases, and remember these hydrogen bonds holding them together. So basically one end of your DNA is upside down in relation to the other. So if this is my 5' end, this is my 3' end over here. If this is my 5' end over here, this would be my 3' end, okay? And one thing that you have to remember as we get into this, it's going to be new nucleotides can only be added to the 3' ends. So when you think about it in terms of that, if you look here, I have a 3' end. I can add a nucleotide right there, okay? I'll be able to add another one over here. I can add my next one right here. There's my 3' end, okay? Now, you need to understand that. So this is, like I mentioned, this is my 5' end over here. This would be my 3' end here. I actually could as well, if I needed to, I could add a nucleotide right there. So just remember the 3 is free, right? So the 3 is free. The 3 is free to add nucleotides. That's the easy way that I've remembered it. So let's get into what you need to know for DNA replication before I let this video play out. So it's actually not terrible. Let's at least kind of get into the introduction here. You want to understand that there's these replication bubbles and they'll fuse and eventually you're left with 2 complete strands. Now, DNA replication begins at what's called that origin of replication. So you have helicase moving in either directions, but The one thing that is important to know is that in prokaryotic cells, things like bacteria, they just have a single replication bubble. Remember that their DNA is circular. So they don't have a linear chromosomes, but the process of all the enzymes, all that's going to work almost identical. But in eukaryotes, we have multiple replication bubbles with these little replication forks. So this one's showing you a eukaryotic chromosome. So you just need to really make sure you differentiate between those two. So when you get into this, we'll show you here that replication fork. Actually, let me show you the clip here real quick, and I'll kind of talk you through each step as we proceed. So this is taken from Mastering Bio. So this one is actually looking in an E. coli, but it's going to work almost the same. This one is only showing you one replication bubble, right? So you have your one replication bubble. If this was eukaryotic, I'd have a replication bubble over here, another one over here to help increase the speed. So let's kind of take a closer look here. And this video does a great job showing you two things. Notice it's opening up on this end as well as this end. So that is basically my replication fork. So we're just going to look at one side here. And you can see a couple of things happening. I have an enzyme opening it up. That's actually helicase. Now, the one thing you want to notice is that these light blue nucleotides are the new nucleotides. The dark blue is like your parental strand. Now, if I paused it, you want to make sure you're understanding this one up here is continuously adding nucleotides. Like there's nothing stopping it. But look over here. I have all of these nucleotides that haven't been filled. This is going to be what's referred to as my lagging strand. So notice I'm adding nucleotides going in the reverse direction. So that would be your lagging strand. So we'll kind of talk about each one of these as the video progresses on. So you just want to make sure that these enzymes here, there's about 7 or 8 that you should be able to understand in pretty good detail of what's happening here. So there's a couple of common test questions here that if we were to take a look at this, I'm adding a new nucleotide right here. So what end of this nucleotide is this? This would be the 3' end. Remember, the 3 is free. So little things like that matter, and it makes life quite a bit easier on you when you can kind of remember that. So, and if I look down here, notice that I can add nucleotides here. So this, if I pause this real quick, that would be my 3' end on this new strand I'm making. So you want to make sure you're clear on understanding that. So this is my leading strand, excuse me, this is my lagging strand because I have to essentially wait. So let's take a closer look at the leading strand as this video is going to kind of take it slow here. So zooming in here, this is my 3' end, that's my new 3' end, my new 3' end. And this is actually, this enzyme adding all of these is called a DNA polymerase 3. So this is just showing you an A pairs with a T, T pairs with an A, G with a C. And so adding the new nucleotides is going to work the same on both the leading and the lagging strands. But it's just showing you that helicase is moving in that direction to the left. I can keep on adding nucleotides in the same direction as helicase, and that's making it my leading strand. So I'm not going to talk about each one of the enzymes particularly besides this. This is helicase, if you didn't know. Helicase is opening it up. This strand up top is my leading, this is my lagging. Let's identify why. So the thing you want to remember is that these little nucleotides in red are actually RNA. So think of it, if you have to paint, you have to lay down a primer. Same thing in DNA replication. Before you can actually add that those new DNA nucleotides, you have to lay a primer of RNA. So that enzyme, if I back that up, that enzyme that added those RNA nucleotides is called primase. And then basically what's going to happen is now you have DNA polymerase 3. I can add those new nucleotides going in that reverse direction. So this would be my DNA polymerase 3 going in that reverse direction, adding nucleotides. Now notice it's going to run into a fragment of RNA again. Now, this is why it's considered your lagging strand, because helicase is going in this direction, but I'm actually adding nucleotides in the reverse on my lagging strand. So I'm going to be running into those RNA fragments. And then these things get referred to as Okazaki fragments. This would be an Okazaki fragment. Now, I have to get rid of this RNA, and it does so in a couple of different ways, okay? So you have an enzyme that's actually going to come through and this is called actually DNA polymerase 1. And this one is actually removing the RNA back with DNA. And that's something that is important. So you're removing those RNA primers and replacing them with DNA nucleotides. So this is actually DNA polymerase number 1. Now there's a really small gap here, and to seal those together, that's called ligase. Ligase is going to help seal those together. So to finish up DNA replication, this is kind of going to show you the whole ins and outs that you're eventually going to be making an exact copy. So let's jump back to the lecture notes and just kind of emphasize what you should be able to know. So kind of getting this through what we talked about earlier is let's at least dial in these enzymes, because you for sure got to know, I like to sequence it. Helicase is the enzyme that actually opens it up. You also have these single-stranded binding proteins that are going to help stabilize it. So basically, if you opened up your DNA, as it opens up, you're going to have these little SSBPs. So these little dots I'm drawn in represent my single-stranded binding proteins. And basically what that does is that helps the DNA from collapsing back upon itself. Because normally when you open up DNA, it wants to close back up. These single-stranded binding proteins, I like to think of them as little tiny pins that keep it pinned open. And then you also have topoisomerase. Now that's kind of interesting. And basically it prevents overwinding. So as your DNA strand opens up, it doesn't want to be strained. So you have these two kind of working. And I notice I have it written a second. I'm saying that these two kind of happen simultaneously. Single-stranded binding proteins keep them pinned open. Topoisomerase, and it prevents any overwinding or overpitching of the DNA helix. So both help protect it. So yeah, here we go. This is a nice kind of overview. You got topoisomerase that prevents any overwinding. These things in silver are your SSBPs, keep it pinned open, and here's helicase. Now notice right here, let's try to identify on a picture like this if this is your leading or your lagging strand, right? So this is my 3' end, this is my 5', the blue is my parental strand. Here's my RNA primer laid down by the enzyme primase. And my question to you is, can I add a new nucleotide right here on this 3' end? And the answer is yes. So I would actually be adding nucleotides going in this direction on this strand. So this would be my leading. Now, let's just say if I added in a primer over here, okay? And hopefully you'll be able to see this now and make it a little more clear. Well, okay, if this is my 5' end, this now is my new 3' end of my new strand. This is my 5' end. So which direction can I add a nucleotide in? Well, remember, just remember the simple rule, the 3 is free. That's the end that can add a new nucleotide always. So I can't add a nucleotide on this side, but I can in this. So I'm going to be adding nucleotides going in the reverse direction as helicase. And that's why this one actually be my lagging strand. So this just kind of reminds you that the 5' end has the free phosphate, the 3' end has the free hydroxide. So synthesizing the new strand, this kind of just reminds you of what we talked about earlier. There's my 3' end. This would essentially be, I don't know, my leading or lagging, but let's kind of just get into key things on this slide that we need to make sure you understand. DNA polymerase, those are going to be the ones that add new DNA nucleotides. So these are the ones that add DNA nucleotides. So those are the ones that actually add in the nucleotides. There's two you have to know. DNA polymerase number 3, that's the main one that we talk about a lot. And then DNA polymerase 1 is going to be the one that actually removes RNA. So removing this, this just kind of reminds you of polymerase 3 adds nucleotides to the strand. DNA polymerase 1 removes the primer of RNA and replaces it with DNA. So let's kind of help you guys identify the leading versus the lagging strand. So if this is my 5' and my 3', and if I'm saying helicase is going in this direction, notice that this is my 3' end of my new strand. So I can add nucleotides in that same direction. Now, if you look at my lagging strand on the right side though, helicase is going from, it's going in this direction. It's going upwards, right? So if I called this helicase, this is my 5' end of my new strand that I'm making, but I can't add a nucleotide here, so I have to add it in that reverse direction, okay? Now, adding in that reverse direction, this becomes the lagging strand, and notice you're gonna run into DNA fragments. You get these fragments of DNA. And those are going to be called your Okazaki fragments. It's DNA in between fragments of RNA. Now, few things here is that in the lagging strand, DNA polymerase 3 moves against the fork. When I'm talking about the fork, I'm talking about this replication fork. And those fragments of DNA are called Okazaki fragments. Now, really two things happen here. You're going to use DNA polymerase 1, and a ligase is going to seal those things together. So let's just kind of show you this. This is what up top is going to be called the leading strand. Again, how do I know? Well, helicase is moving in this direction. Notice that I can add a new nucleotide right there, so that would be my leading. But when I look at my lagging strand, notice here's my 5', 3'. I add DNA nucleotides in that direction. And then it shows you DNA polymerase 1 is going to, DNA polymerase 3 is the one that adds in all those nucleotides. But when you get down here, DNA polymerase 1 is the enzyme that actually removes that RNA. So what is the function of DNA polymerase 1? It removes RNA and then it replaces it with DNA. And it's really, really tough to see even in your textbook, but there's a small gap. Ligase is going to help seal those gaps, okay? So let me jump back to the video and just show you again on the lagging strand, because there's a lot of moving pieces here. And so when you look at what is happening with the lagging strand, let's check this one out. This is primase. Primase is going to lay down that small little section of RNA. There's my 3' end, right? DNA polymerase 3 is essentially going to be adding those new nucleotides going in the reverse direction. So I'm adding those new nucleotides again and again and again and again. So this is basically the creation of an Okazaki fragment because I have DNA in between these RNA fragments, okay? Now I got to get rid of this RNA, so hopefully this will make some sense now. To get rid of this RNA, this now is called the DNA polymerase 1. It removes RNA and replaces it back with DNA. So this would actually be considered my DNA polymerase number 1, okay? So that's going to be the main function of this enzyme. It removes RNA, replaces it back with a DNA. That's called DNA polymerase 1. Now the last thing, to get rid of this tiny, tiny gap here, I seal it with the enzyme called ligase. So a lot of moving parts on the DNA replication, but it all happens insanely fast. Now, one final thing about DNA replication that you want to at least make sure you feel comfortable with is the proofreading. DNA polymerase actually proofread it, so that it always is going to go back and double check if there's any errors. It's going to fix it. You're going to repair it, and any damaged DNA can be replaced. Now, DNA can be damaged if there's any harmful mutagens, things like UV rays, if your DNA gets methylated. So in terms of that, we have nuclease excision repair, where an enzyme called nuclease cuts out and replaces damaged stretches of DNA. So let me show you what that looks like. If you get kind of a stretch that's bad DNA, not just a mismatch, but it's bad, faulty sequencing, nuclease is an enzyme that actually cuts out the bad one. DNA polymerase is going to add the correct one, and ligase seals it. So it's really, really cool. Your DNA proofreading is very, very specific. So that's pretty insane. So a nice little overview here of a leading versus lagging strand. Helicase is going in that direction. And it's just kind of showing you that this is my leading strand because I can keep on adding nucleotides. This would be my lagging strand because I would be adding nucleotides. And remember, the 3 is free, adding those nucleotides in the reverse direction. So always just kind of quiz yourself. How is this RNA going to get removed? Well, DNA polymerase number 1. There's going to be a tiny gap. Ligase seals it. So last thing is these telomeres. This is going to be typically with eukaryotes only. So everything I've talked about for the most part has been both eukaryotes and prokaryotes. But I do need to emphasize that these telomeres are really going to be in eukaryotes only. Okay, so just to remind you, what are these eukaryotes? Cells with a nucleus, linear chromosomes. So basically, you have these protection caps of A's and T's, adenines and thymines. Okay, now think of this as like the quote-unquote good DNA, meaning that this is the DNA that you actually need to read and express to make your proteins. But every time you replicate your DNA, these telomeres get a little shorter and shorter and shorter. because every time it does mitosis, you lose some at the very, very end when it replicates. And there's contributions on this to aging. So telomerase is the enzyme that adds telomeres to gametes. So basically there is an enzyme that actually adds these telomeres, but for some reason, that enzyme, telomerase, where these telomeres are being added, is only active in gametes. So once a newborn baby is, viable and actually given birth to, telomerase no longer functions. So there are some crazy studies happening, things like lobster, their telomerases are always active. So they don't really die of old age. They die of natural causes in the sense of like either they're eaten or something else happened to them, but they don't actually die of old age. So all kinds of crazy genes out there. Now, this is kind of a nice little overview of all the enzymes that we had mentioned in this video. So do make sure that you spend some time and feel comfortable with what each one of these is doing. So overall, that in a nutshell is kind of the basics of DNA replication. I'm going to play this last little quick clip to remind you guys of what DNA replication looks like. So remind you guys that the whole end goal of DNA replication is to make an exact copy. So looking at these bubbles, how they'll fuse together matters. So test yourself like, okay, what is this enzyme? It's helicase is opening it up. This is my one replication fork, okay, right here. So there's that first enzyme that's going to happen. Now, test yourself, okay? What has to happen first? I have to lay down a primer. Even on my leading strand, I have to lay it at least once, okay? So I'm going to lay down my primer of RNA. So that's my 5' end. This will be my 3'. So if I can add a nucleotide right here, this is my 3' end, again, hopefully you can understand, okay, well, what enzyme is this now? Well, it's DNA polymerase, and it's actually polymerase 3. DNA polymerase 3 is going to be the one that's adding in these new nucleotides. A pairs with the T, T pairs with the G, so on and so forth. So this is going to be the building of it. So keep in mind, your 3' end is growing. I can always add on that 3' end. This is my 3' end. This is my leading strand. I can continuously add these nucleotides. And notice I'm adding these nucleotides in the same direction as helicase. Now notice my lagging strand, that would be my 5' end. I can't add a nucleotide here. So in terms of this, it basically makes chunks of DNA. Chunks of DNA is my Okazaki fragment. So I have to wait for helicase to open it up. I'm going to lay down my primer, and then I'm going to add nucleotides in the reverse direction. So that's my 5'. This is going to be my 3'. Remember, the 3 is free. I can add nucleotides in that 3' direction. So letting this play out, continuously adding those nucleotides in that direction. Beautiful. I have to lay down a primer again. And hopefully you're noticing that you have to lay down primer a lot in your lagging strand. So just kind of goes to show how complex this whole process can get. Now, I have all of these little primers. I need to actually remove it. Your book calls it DNA polymerase number 1. So DNA poly 1 is removing it. Don't worry about hexanuclease. So DNA polymerase 1 replaces it, and then ligase is going to seal it. And so it just shows you that the enzyme ligase seals any gaps that you had. And that should wrap up your 2 new strands of DNA. So again, you're making exact copies, each with the half old parental strand and half of the new parental strand. So that is DNA replication and chapter 16.